is Chapter 7, Section 4, and it's on human genetics and pedigrees. The key concept here is that a combination of methods is used to study human genetics. So human genetics follows the patterns seen in other organisms. The basic principles of genetics are going to be the same in all sexually reproducing organisms, meaning that we can study things like flies, and what we learn about their genetics we can still apply to human genetics because every living organism shares the same basic genetic code. They each share DNA, where DNA has the same base pairs A, T, C, and G that are in all organisms. And in ways that genetics works, especially in eukaryotic organisms, organisms that have a nucleus like us, principles of genetics that apply in those organisms still apply to us. And so, because doing experiments on humans is unethical and dangerous, and no one wants to be the human subject in those studies, we use what are called model organisms or other organisms and study their genetics and apply what we find to ourselves. It's important to note that a lot of inheritance in many human traits is complex, as we mentioned in previous sections. However, single gene traits are important in understanding human genetics. So some traits are controlled by one gene, other traits are controlled by multiple genes. Okay, so talking about sex-linked genetic disorders. Females are going to be the carriers of these genetic disorders. And the reasoning for this is just getting back to what makes a female, and that's having those two X chromosomes. Whereas males have an X and a Y chromosome. So say you have some sort of genetic disorder that happens on the X chromosome. For males, you only have one X chromosome, where for females, you have two. Meaning that males who say they had some sort of genetic disorder that caused Duchenne's muscular dystrophy, DMD. If it happens in a male, that's for sure going to express itself because the males don't have another copy of that X chromosome to make up for that missing gene or that dysfunctional gene. Whereas females are going to have two copies of their X chromosome. So, say this X chromosome has the mutation for the muscular dystrophy, they still have another X chromosome where they might get enough production of the gene on the other X chromosome in order for them to still survive and reproduce and have offspring. Whereas for males, this muscular dystrophy might prevent them from becoming adults and having offspring. Males are going to express all of their sex-linked genes, meaning that they aren't going to be carriers, as I mentioned. But also, say it was something that happens with a Y chromosome, same thing applies. Males only have one Y chromosome. So if there's some sort of genetic disorder that's based in the Y chromosome, that's going to be, you're not, in other words, if you're a male, you either are going to have the disorder or you're not. You're not going to be wondering, oh, do I carry, am I some sort of heterozygote that has, that's carrying one of those recessive alleles, because you have one of each of the sex chromosomes. Whereas since females have two X chromosomes, they can have that recessive allele that's kind of hidden in the background and not being expressed. Okay, so bring up the idea of a pedigree. A pedigree is just a chart that we use to trace genes in a family. And so a pedigree, if we jump down to this right here, it looks kind of complicated, but they're really actually pretty easy to read once you get used to kind of what all the symbols mean. Coming up here from the top, this is the parental generation. So you have a box means a male, and then a circle means a female, and then the F1 generation is going to be their children. So the line that comes down here and extends down and down and down are going to be the children of these parents. So they have 3 children, 2 female and 1 male, and then this line sticking out here from the male and here from the female represent their spouses. And so then they can have their own children where they have 3 and then 3. The first generation of kids is called the F1 generation, and then the second generation is called F2. Whenever you see, so for the males, whenever you see a clear box, one that's not shaded in, that means that they aren't expressing that phenotype, or in other words, they don't have that trait. In this case, the trait is widow's peak, which widow's peak, um, if I go all the way back to the first slide, widow's peak is what this gentleman has. It's that little point at the front of the forehead, and that's actually known as a dominant trait. If you remember back to Mendelian genetics, a dominant trait is where you only need one of the chromosomes in order to express it. Whereas a recessive trait is something where you need 2. You always have to have 2 of the alleles in order for the trait to be present. So this female right here is shaded in, and she has the widow's peak, but she's going to be a heterozygote. If I rewrite heterozygote, it's something we've talked about previously, and that's just something where you have, it's an organism that has one of the dominant alleles, the capital letter, and one of the recessive alleles, the lowercase letter. Whereas this, the male right here, is a homozygous recessive. Homozygous, homo meaning same, and then recessive, I won't rewrite recessive, but recessive referring to the lowercase letter. Okay, so going to their individuals, their offspring, you see this male here shaded in, so that means they have the trait widow's peak. This female right here doesn't, and this female does. Oftentimes, what happens in these problems is sometimes they won't show you the children. Maybe they'll just have like an X right here, and they'll ask you, "Hey, what does this X stand for? What is the genotype this X stands for?" And how you would determine that is by looking at the parents and looking at, the symbol here. So we know that this individual, this female, has the widow's peak, has that phenotype, and then we're going to know that they're going to have to be a heterozygous, capital W, lowercase w, because there's only one capital W in the parents. And if you remember, our Punnett square, so if we put the parents up here, capital W, lowercase w, lowercase, big W, little w, little w, little w, big W, little w, little w, little w. None of the offspring have 2 capital W's. So that removes the only other option for what this genotype could be. Okay, the other type of pedigree is going to involve sex-linked genes. And so if the phenotype is more common in males, that's going to be your giveaway for the gene being sex-linked. And the reason for that is what I mentioned in the previous slide, where males only have one of each of the sex chromosomes. Specifically, they only have one of the X chromosomes, whereas females will have 2 copies of the X chromosome. And those 2 copies of the X chromosome for females allows them to have this recessive allele here, where they can be a carrier of the disorder, but they don't actually express it. So whenever you see a half shaded symbol here, that means that this individual is a carrier, but they don't actually show that phenotype. And the phenotype here is color blindness. So this female is a carrier for color blindness, but isn't actually showing that phenotype because she still has one normal functioning X chromosome. An interesting thing here is number 5 here, this female. We don't know whether this female is going to be someone who's doesn't have color blindness, or is going to be someone who doesn't have color blindness but is just a carrier, because we don't know whether, we know that she got this capital M from her dad, because the males are always going to donate their X capital M. All right, so if I do this, if I do the Punnett square real quick, let's put the dad up top. big M, Y, oops, I'll put the female right here, X little m, X big M, Y, X big M, X big M for female. Then down here you got X big M, X little m, and then X little m, big Y, meaning that, meaning that you can get an individual who has both of these X chromosomes with the capital M or one of the lowercase m's. We don't know which one of these offspring it's going to be. So that's really, figuring out these pedigrees. It's just a matter of going off of what you know and kind of starting from these parents and thinking what they have to be or what they are. So I'll give you guys some worksheets to kind of help you practice them, because that's really the best way to understand them. But as long as you understand what all these symbols stand for, you can typically kind of, just using some basic logic and common sense, can figure out what the unknown ones are. Okay, so almost done. Going over a karyotype, which we've talked about before, there can be several methods that, we use a karyotype as a picture of all the chromosomes in a cell. And this is one of the methods that we use to help map human chromosomes. So just to review the karyotype, you got, if you it's got all 23 of the human chromosomes, where if I jump to, say, this one right here, you get 2 homologous chromosomes. Remember, homologous chromosomes have the same genes. That's what those banding patterns are showing, where you got one from mom, one from dad, and so on, and then XY, that's going to be a male. And really what we use karyotypes for is to show the changes in chromosomes, where these changes can be maybe the deletion of a part of a chromosome, or you completely lose a chromosome altogether, or as in the case here, you gain an extra chromosome here at number 10 and number 4, and any other sort of large change in chromosomes, or maybe you're missing. So if this shading is representing just this entire, like half of this chromosome arm just going missing. So we use karyotypes. Actually, this is what you got right here. Number 1 is the example of it missing. Karyotypes, they just show the pictures of all the chromosomes in a human. You can use them to look and see what is it. You know what normal looks like, because you have to have 2 of every chromosome number, and you look and see where are we missing something at. Or maybe it's normal and there's nothing wrong with the baby, and then that's great. Okay, so I'll do it for this section.